Work, yeah. So hello everybody. Hope you enjoyed your holidays and had the chance to recharge for 2024. And as Sylvian Fakel said, I'm here today to talk to you a bit about what has been my research interests in the past, and also since I'm a new member here in CBU, and also a very brief overview of what my role will be here in working with Sylvia. And so I chose this title because it more or less captures what are my interests and the hypotheses I've been testing in taxonomic diversity across space and time. And yeah, my name is Matthew Moreira. I'm a recent PhD. I did my PhD in a joint program at the University of Aveiro, but also with the University of Lisbon in 2023. And yeah, that's the introduction. And so let's begin. I will begin talking about a bit of about biodiversity. This is not homogeneous over space or time, and it's generally described under three types of species richness patterns. The first one, the spatial species richness patterns, describes the overall discrepancies that we find in by the species richness among regions, and this in some groups we can find that the species richness decreases from the equator to the poles, the so-called latitudinal biodiversity gradient. But other groups, we can also find that species richness accumulates in certain regions on the globe, the biodiversity hotspots. There are also the clade-based species richness patterns that describe the discrepancies in species richness among clades in the tree of life. For example, we have the phylum Placozoa, which is monotypic with one species, compared with, for example, the phylum Arthropoda with more than 1 million species. This can also be verified in flowering plants, in lepidosaurs, or even in mammals, where we find groups with very few species or even monotypic compared to thousands of groups of species in their sister groups. A third type of species richness patterns are the now called trait-based species richness patterns that describe the discrepancies we find when we identify that more species have, for example, state A than state B of a given character. This can be verified among regions or clades, but usually you can see that there's more species with a state of a character than the other. An example is the evolutionary paradox of sex. It's a puzzle for evolutionary biologists to understand why more species we find more sexual species than asexual species, despite the overall costs of being of sexuality. And so then these types of richness patterns have led many ecologists and evolutionary biologists to one central question, to why certain clades or traits in the tree of life and different regions on the planet have outstanding taxonomic diversity, while others remain species poor. And this is more or less my main motivation until this point, to identify traits that can help us understand better these patterns. And so there are several direct causes that have been identified that can directly influence species richness patterns. For example, in spatial richness, this is directly influenced by speciation or extinction, dispersal frequency, or even the timing of colonization among locations, which will dictate how much time species have had to build up. Similarly, in clade-based richness patterns, we also find that clades with more species are usually, they either have higher diversification rates or they are older. And also influencing trait-based richness, the same, we have diversification rates. This is a state A, for example, if it's more common than B, it will be linked to higher diversification. The trait origin frequency can also influence this. These patterns, for example, if state A is lost less frequency, so the transition from state A to state B is less frequent, but also if state A is more frequently gained, so there are more transitions from state B, let's say, to state A. Also, the age of a trait can also influence trait-based richness patterns. So an older trait will be more common under this hypothesis. And so 3 general hypotheses have been formulated. 2 are common between all of these richness patterns, the age hypothesis and the diversification rate hypothesis. And also a 3rd hypothesis involves the transition rate, which is common between the spatial richness and the trait-based richness patterns, either through dispersal from a region to another, or even from character evolution, as we've seen from trait origin frequency. So the age hypothesis predicts that regions, clades, or traits with more species are older and have had more time to accumulate species richness. That the diversification rate hypothesis predicts that regions, clades, or traits with more species have faster rates of diversification, accumulating species at a faster rate. And then the trait transition rate hypothesis predicts that regions or traits, in this case A, with more species will have more transitions to this region or a certain state, and or less frequent transitions from that region or state. And so these are the direct causes of species richness. There are many other indirect factors that will have to act through the time, diversification, or even the transition, the frequency of transition from those regions or traits. And yeah, and so, but what causes these, what are the evidence we find for each of these richness patterns? So regarding the spatial richness patterns, it has been found that colonization time drives spatial richness patterns over shorter timescales. At least this is a study for amphibians that found evidence for the age hypothesis for amphibians in the Andes, where they found that within the families and within the Andes, that species in different elevational zones, you can find a positive relation between the colonization time and species richness. So older elevational zones will have more species, but then they also found that diversification rates drive spatial richness patterns over longer timescales for the same group. And there they divided the regions not by elevational zone, but among families and among biogeographic regions. And they found that diversification associated with the Andes explain better the species richness, the higher species richness in this region. As for the evidence for the clades-based richness, the relationships between richness and age were weak or negative, so not in support of the age hypothesis. Here we have the relationships between richness and the age for among the clades of the same rank, so families, orders, classes, or phyla. But they found that stronger and positive relationships between richness and diversification rates in support of the diversification rate hypothesis, explaining clade-based richness, where we have, for example, in families, that the families with more species will tend to have higher diversification rates and are not particularly old. And they found this pattern for regardless of the rank, sorry. Then, regarding the trait-based richness, evidence is mixed. 15 studies out of 25 show that the oldest state was a prevalent state in support of the age hypothesis, so older clades tend to be more frequent. But there are other studies for other states that show that faster rates were associated with this prevalent state in support of the diversification rate hypothesis. So here the support is stronger for the age hypothesis, meaning that usually more common states will be associated with, will be older, but there are also cases in you find that diversification will be associated with more prevalent traits. And so, but then if diversification and alongside time help explain the variation in species richness patterns, what helps explain the variation in diversification rates? Species traits, as we've seen in the previous slide, can have a strong impact in diversification. There are many species traits that can influence diversification, either climatic niche, physiology, body size, or even reproductive mode. And these traits will either hinder or promote diversification. Remember that diversification is usually calculated as speciation minus extinction. So a trait that will promote diversification will either promote speciation and or hinder the extinction, and vice versa for if it hinders diversification, it will hinder speciation and promote or promote extinction. And this will be revealed. This is a very simple example, but usually we'll find if a trait promotes diversification, you'll find young and species-rich clades. But if it hinders diversification, you will find old and species-poor clades. And this has been more or less my focus, to identify traits that can influence diversification to better understand species richness. I have been usually focusing on terrestrial vertebrates, but with a stronger focus on squamates. These include lizards, amphibians, and snakes, which are diversified early in their evolutionary history to occupy most of their adaptive roles we observe today. And they are sister to Ataras, which are from the group Rhicocephalus, that is monotypic with only one extant species. This group has occupied almost every region of the planet. It's the most diverse group of non-avian reptiles, rivaled only by birds among terrestrial vertebrates. It has high phenotypic and ecological variability. They have adapted to different habitats and diets, and very rare sexual phenomena has occurred in its clade. And so it's an exceptional group to study species richness patterns. Now I will focus more on three types of studies that myself and colleagues have conducted under this scope of taxonomic diversity, which can be grouped in three types of studies: the trait-dependent diversification, reconstructing character evolution, or the macroevolutionary scenarios of trait evolution. So one very simple and straightforward way to test the role of a trait on diversification is by establishing a link between a trait and diversification to address how a trait influences the variation in diversification. To identify is trait-dependent diversification. This includes the tip rate correlation methods, which will, in this case, is the most famous is probably PGLS, which will basically detect if there's a relationship between diversification and a variation in a trait. Don't forget that diversification is the speciation minus extinction. But more recently, ESM has been proposed because it's faster and has simpler assumptions than PGLS. But more importantly, it is a simulation-based approach that allows to test for this in a very quick way. And we did, in this study, we went a step forward and we introduced CSM-GLM, a multiple regression trait-dependent diversification approach, because we wanted to test for not only one trait, but the coupled effect of two traits on speciation. And we developed this method introducing GLM. That's why we called it CSM-GLM, because it uses generalized linear models to allow to test for the effect of multiple traits on diversification in a fast and robust way. Overall, we show that the CSM-GLM has very important properties of tip correlation methods. It is fast, taking less than 25 seconds for trees with 1,000 tips. It is robust with strong power, even with incomplete taxon sampling. It has high power, especially for moderate and large-sized trees. And it also showed low false discovery rates. And by using ES in GLM and the family Leolamidae, which is a family of lizards that radiated in the Andes, we were able to show the coupled effect of both body size and geographic range size on explaining 19% of the variation in speciation rates. And we show that larger species coupled with smaller range sizes were linked to faster speciation. And this can be largely explained by this genus Fimaturus, which you can tell that has faster diversification, speciation, coupled with relatively larger body sizes and also smaller geographic range sizes. And so this is a very simple and straightforward way of testing the role of a trait on diversification. Alternatively, we can be interested in asking what happened with a given trait to address its current distribution or even its relationship with other traits. Under this scope, we addressed a large-scale evolution of body temperatures across land vertebrates. Body temperature is a very important trait for all animals, and we were interested in answering fundamental questions that were unanswered until this point at this scale. That include, for example, that if endotherms have higher body temperatures than other tetrapods, if endotherms have more constant body temperature among species revealed by lower rates of evolutionary change than ectotherms, if day-night activity patterns help determine body temperature among species, or even if body temperature is evolutionarily conserved among species. Regarding the endotherm and ectotherm divide, we find that there is no significant differences between body temperatures. We found no strong differences in mean body temperature, largely explained because birds and mammals do have higher body temperatures here on the right, but there's much overlap between lepidosaurs, which include all lizards and snakes. There's a lot of overlap here with mammals. And also, we find that amphibians, although they have lower body temperatures, there's a huge variation and they also overlap a lot here in the middle. So we found no differences between endotherms and ectotherms body temperatures. We did find that they are more similar within each other than ectotherms. Endotherms show low rates of change relative to ectotherms, which include these four groups, especially with amphibians that show the highest rates of change in body temperatures. We find that clade level rates of TB were significantly slower in endotherms, meaning that they are more similar between each other. And also that, as we've seen before, that TB body temperature in endotherms is not strictly higher than ectotherms, but they are more similar, revealed by the lower rates of change among species within clades. We found also that body temperature is tightly linked to day-night activity patterns. We show that for both groups, diurnal species have higher body temperatures than nocturnal, which is kind of intuitive for ectotherms, because by night they cannot achieve higher body temperatures. But this different we were not expecting for endotherms, probably because they are thought to maintain similar body temperatures across environmental conditions. And one potential explanation we suggest had to do with energy requirements, and probably this one way of explaining it is because it's easier to maintain cooler body temperatures at night by nocturnal species, but also it's easier to maintain hotter body temperatures for diurnal species by day. This was one of potential explanation for this pattern. And also, we also find that this trait is largely conserved over deep time scales. This is we found strong phylogenetic signal even at the scale of 350 million years old. In contrast to the idea that conservatism disappears over deeper timescales. You also find that many turtles, sorry for the yellow on white, lepidosaurs and amphibians have maintained body temperatures similar to the value estimated for the crown group or ancestor of tetrapods. So this has been largely conserved and probably reveals physiological limitations. The last group of the last study I wanted to show you had to do with macroevolutionary scenarios of trait evolution, where we could be interested in identifying the factors that shape the prevalence of a trait that we observe nowadays. And in this study, we were interested in understanding the role of parthenogenesis as a self-destructive trait. And this because going back, asexual reproduction, although it occurs in vertebrates, there are 4 groups of asexual reproduction. And what distinguishes parthenogenesis from these other 3 is that it does not require male fertilization for egg formation, and it is only identified solely in lizards and snakes among vertebrates, where you can find other types of asexual reproduction also in amphibians and fish. And this is an extremely rare trait. At the time of this paper, there were 39 out of more than 11,000 species described as parthenogenetic. And so what could explain this unstable macroevolutionary pattern? We asked if it has to do with increased extinction rates or increased rates of reversal, for example, from asexuality to sexual reproduction, or even a combination of the both. One explanation is this is a self-destructive trait. These are traits described as arising often, but being coupled with higher extinction rates, leading to short-lived species and evolutionarily phylogenetically sparsely distributed. But also they arise often, but are lost very fast with high rates of reversal, the loss of the trait. And parthenogenesis could be considered a self-destructive trait because, as you can see here in the phylogeny, this is phylogenetically scattered, and it's extremely rare trait among vertebrates, let alone squamates. And so this instability observed suggests that it either increased extinction rates or increased rates of reversal, or a combination of these 2 factors. So we went on to test this, and we did a set of forward simulations of macroevolutionary scenarios, where we constrain a combination of speciation, extinction, or transition. And it was simply we did these nine scenarios, where, for example, in the first, we did not constrain these rates, and we simply simulated a tree with a binary trait, constraining either in this scenario we make the tree have the same speciation, same extinction, same transition rates between the two states of this character, and this for several scenarios. And then we calculate a set of 3 metrics that describe different characteristics of a phylogeny, either TARS, Noto, SSCD, and FPD. And then we can identify if our observed pattern is similar or not to these simulation scenarios for a lot of iterations. This is what we obtain. This is the distribution of the values for each simulated macroevolutionary scenario, and this dashed line is our observed value. And then we can reject or not this given scenario, and we get to the conclusion that parthenogenesis is self-destructive because we do not reject any model that identify our extinction rates relative to speciation. So parthenogenesis in this case would have higher extinction rates and lower speciation, or even scenarios where high rates of reversal were identified, which is C and F. I will not get too much detail. And then we reject the models that identify higher speciation rates relative to extinction. So, for example, if the trait is gained very fast, we reject these models. So we go in even further as to suggest that parthenogenetic clamates increased extinction rates are responsible for their rarity, because reversals from asexuality to sexual reproduction is extremely difficult. And so this would not be, although it can create similar scenarios of trait evolution, because we do not reject models that have high rates of reversal, it will be extremely difficult due to physiological limitations to this reason of reversal from asexuality to explain this pattern in parthenogenesis. And so ultimately we identify parthenogenesis as self-destructive due to its negative relationship with extinction. And so yeah, this has been my work. I've been trying to identify traits that are related to diversification and that help explain these different species richness patterns. And now I will be working under the project PLACES, which stands for Planning for the Conservation of Life on Earth in the Light of Evolution and Sustainability. And my main role will be to identify the determinants of interspecific genetic diversity across taxonomic groups and regions. And these can be either evolutionary, geographic, environmental, or functional traits, or a combination of these traits. And ultimately, this project will aims at optimizing biodiversity conservation strategies by incorporating evolutionary structure in future conservation agendas. And this is more or less what I can tell you right now, because I'm here just for like 30 days. So yeah, hopefully my skill set will allow me to achieve this. And feel free to, yeah, thank you. And if you want to check more about my work, you can check my website. And yeah, thank you for the attention.